All right, I'm Connie Lee. Most of you know who I am already, and I am really excited to be introducing tonight's webinar. We have one more person who's going to be presenting a little bit. So what I wanted to preface with first, though, was a few housekeeping items. When you have questions, please use the Q&A. You'll see that at the bottom of your screen, there's a Q&A button rather than the chat. That allows us to do a better job of curating the questions and making sure that they get asked. Also, at the very end, if you check the chat, you will see a link to our support group. We have a weekly support group every Thursday. It happens right after this webinar is finished. And so if you click the link, you'll be able to join and probably continue the discussion between you. So without further ado, I am here with what seems like a very large team from the University of Chicago. And I'm going to let Dr. Awad introduce his team. I will introduce him. Dr. Awad is the University of Chicago's John Harper Seeley Professor of Surgery. He leads the Neurovascular Surgery program and co-leads the Stroke and Neurovascular Care team. He is the director of our CCM Center of Excellence there, and he's also the chair of our Scientific Advisory Board. Dr. Awad, please take it away.
Thank you so much, Connie. I'm really excited about this attendance, and you know, we have talked about this problem of aging and the cavernous malformation for the last 3 or 4 years with a lot of interesting angles on it. But aside from what the science tells us and how the questions are asked, everybody worries about aging, and when we have a medical problem on top of it, it becomes a serious concern. We'll try to address some of it this evening with you. I want to first introduce the 3 people who are with me, and I'll call on them for some brief comments as we get to particular aspects of the webinar. So first of all is Agnieszka Stadnik, whom many of you already know, and she leads our clinical research effort at the CCM Center at the University of Chicago, and that is no small task. She handles more than half a dozen IRB-approved projects, most of them centered on the cavernous angioma or on bleeding in the brain. She interacts with a lot of the patients. And the way we generate knowledge on human disease is to do human subject research with consent and participation. And she's the point of contact with all of you when you come and see us in this regard. 2 other scientists do a lot of work related to the aging brain, and I asked them to join me tonight. Dr. Girard is actually the research assistant professor with our cavernous malformation research group. And for those who do not know, behind the scenes, he's the one that identified the role of vitamin D deficiency with the activity of CCM. So he was the first to show that. Later, he led a huge effort that has allowed us to identify molecules in the blood that correlate with the bleeding of the cavernous angiomas. And many of you who come to the University of Chicago have given your blood samples for this very important research. Part of the reason I asked them to join us tonight is that about 3 or 4 years ago, we decided to look at the common gene abnormalities that affect our brain as we get older, and see what is in common between them and the cavernous angiomas. And you might wonder, why would anyone want to do this type of work? And we'll tell you the reason for asking those questions. He did a lot of studies in looking at the biomarkers of the brain as we age, and as we start to have problems with bleeding in the brain as we age. And there is a theme of commonalities between what happens in cavernous angioma and in the aging brain that we'll talk about. And he has with him his daughter Lorella, who we may see the top of her head. Is that right, Romuald?
Oh, I'm so sorry. I didn't realize that you could see her.
That is okay. This is a family-friendly crowd, so we are all fine. And Abhinav, whom you see also with us, is an MD PhD student in my lab, and Abhinav's work is focused on the bleeding in the brain that occurs in aging. So, and it will make a little sense why a cavernous angioma lab is studying this in a little bit, and I'll ask him to also say a few words about his work. So, in general, there are basically 4 general aspects of how cavernous angiomas interact with aging, and I think they probably account for almost all the questions you have on your minds, because you ask me some of those questions when I see you in the clinic all the time. So the first thing that comes to mind is, does the presence of a cavernous angioma, solitary or the familial disease, impact our brain as we age? Does it make us more likely to have cognitive problems, more likely to have age-related bleeding, and more likely to have other problems with the brain as we age, just because we have a cavernous angioma? The second question has to do with the genes that are impaired in cavernous angioma and the genes that are impaired in aging. So as we age in our brain, there are groups of genes within the brain that starts to get disturbed as a result of the aging process. And I'm not talking here about genes that you give to your family members, the germline genes, but I'm talking about the genes in the brain itself. And we also have groups of genes that are impaired in the brain of familial cavernous angioma patients, and also those with sporadic disease. So how do those genes interact with each other? And does one process make the other worse? The third maybe a dimension of the question has to do with iron in the brain and what this does to aging and cognitive changes, and whether this happens in regular aging, and how the iron that's present in cavernous angioma can make that worse or better, and so forth. And the iron also relates to another aspect called permeability. So permeability means how leaky are our blood vessels in the brain. So normally, blood vessels in the brain are extremely tight, meaning they don't leak. They're structures that are delivered to deliver blood, deliver oxygen and nutrients to the brain without having any leakage. And this impermeability, if you would, is called the blood-brain barrier. So it's a very important concept in biology. There are cells and other structures that make the blood vessels absolutely tight in terms of not leaking. So what happens if we have genes in our brain that make our blood vessels leaky? As it turns out, cavernous angioma genes cause leakiness in the blood vessels. And what does that really do to the brain, and what does it do to the brain as we age? And so that's the third dimension. And then the fourth dimension has to do with the plasticity of the brain as we get older. So we all get injuries and we bounce back from them. We have medical problems, we bounce back from them. But we all realize that you bounce back differently when you're 30, 40, 50, 60, and 70. so that as we get older, the whole ability of the brain to accommodate injury and to deal with disease becomes a problem. And if you have a burden that cavernous malformation has caused to you from prior strokes, prior brain surgeries, the sum total of that burden can be significant in the aging person, more so than in the young patients. So those are kind of the general areas that I want to address before we get to the questions and answers. So the first thing that I want to talk a little bit about, and then I want to give a chance to Romuald to say a few things about what he found in this regard, is forget cavernous malformations themselves. As the brain ages, do we get more leaky vessels in our brain as a result of aging? And also, do we have more iron in the brain that gets deposited as we get older, regardless of having cavernous angioma? And the final thing is the aging brain has within it little bleeds. And people get these bleeds mostly as we get older. They are a little bit more in people who have high blood pressure, but they are also a little bit more in people who have a problem called amyloid in the brain, blood vessels, and that makes the blood vessels leak. And when they cause these microbleeds, it becomes a problem to tell the difference whether this is a cavernous angioma or a microbleed. And there is not 1 week in my clinic where I don't have 1 or 2 patients where this is even a problem for me to tell the difference, even though I know both problems very well. So the appearance on MRI looks like multiple bloods, some bigger than others, exactly as you see with familial cavernous angiomas. And this happens in the brain of people who don't have the CCM gene. And we call this cerebral microbleed. And this problem of microbleed not just looks like cavernous angioma, but may have some of the same triggers that makes those microbleed happens as cavernous angiomas. So let me first maybe ask Romuald, if he doesn't mind, to tell us a little bit when he studied the genes of the aging brain and looked at the groups of genes that happens in the brain as we get older. And we published this in a paper that he authored in Giro Science about 3 years ago. Romuald, what were the general things that you found?
So, yeah, sorry. So obviously, as you know, all the team know, that was kind of a weird question. Even Connie, when we presented the data, was kind of surprised of seeing this kind of comparison between CCM and the aging brain. But what we found, we found that some of these common genes were mostly associated with how the vessel become permeable. Like how come it's easier for the blood to go outside the vessel. So that was one of the first very interesting finding. But the other one was also everything that is associated with the inflammatory pathways. So basically, even when you age, that's something that is known. When you age, you have an increase of your inflammation all around the body. But this inflammation is also common with what we see in CCM disease, in the CCM lesion. So that is two very important components that we are studying with CCM disease were also found in a patient, in healthy patient, but older healthy patient. So as also Dr. Awad knows, we don't really say a cut age for older patient because people doesn't like that. But basically, we did.
It's younger than me, then it is young.
So let's say higher than 70 years old. So when you are older than 70 years old, you end up to get this leaky brain with a kind of inflammation around that may, I say may right now, be predisposing to actually a bleeding, a stroke, a kind of a massive stroke. So that's what we found, and that's what we are actually studying right now with Abby, to see if this commonality can help us later on to find some biomarker and see how actually patient with CCM and becoming old can, sorry, so that's my little girl that is saying that it's a super project, that actually how this biomarker can be used, or how this biomarker can become actually only on one case, either older patient, either CCM patient.
Yeah, so when we talk about biomarkers, these are the molecules in the blood that we will plan to be assaying for, that will tell you in some ways how is your brain aging. So if we have more of these molecules, then that will tell a little bit that the brain is aging more or aging less, or aging more leakily, or aging with more inflammation. But the real worry that we have, and this is what Abhinav is studying, is this predisposition to hemorrhage. Of course, all of you live with cavernoma and you worry every day about, is my cavernoma going to bleed? But what you don't realize is that the aging brain after age 60 or 70 starts to have a very similar predisposition to hemorrhage, with about 0.5% per year risk of bleeding. We call that a hemorrhagic stroke, and it's not a cavernous angioma, but it is a result of years of weakening of the vessels in the brain with aging. So the groups of genes that are different in the aging brain were surprisingly similar to the groups of genes we find in cavernous angioma. So I'm going to mention a few molecules that some of you, the more savvy people in the group, have already heard these molecules. So you've all heard about Rho kinase, you know, because we're ROCK inhibition and so forth. So Rho kinase is a enzyme that makes the blood vessels leaky. When there is Rho kinase activity, there is leakiness. And we found that to be significantly increased when we lose the CCM gene. So in the cavernous malformation lesion, where the CCM protein is lacking and the CCM gene is not expressed, there is a huge activity of Rho kinase that's causing the blood vessel to bleed and to be a cavernoma. Exactly the same way, the endothelium of the aging brain acquires a slower but definite increase in Rho kinase activity. So it happens, it's like playing slow over time, over many, many years. But the amount of activity of Rho kinase in the endothelium of the aged brain ends up getting very close to that we see in familial CCM disease. So when we measure that, it is the same. That also means that as our brains get older, they become a little bit leakier, like Romuald mentioned, and we in fact have a project funded by the NIH where we're studying the leakiness in the human brain with aging in a similar way to how we studied it in cavernous angioma. And to remind you, what we found in cavernous angioma is that if you have a solitary lesion, that area is leaky, but the rest of your brain is not leaky for you are exactly as the other people your age. That's because you only have a mutation in the area of the lesion. But if you happen to have the familial disease, as I'm sure many of you on the call have, every cell in the body has half as much of the CCM protein and half as much of the CCM activity. That's not enough to cause a cavernoma unless you lose all of this protein. But if you happen to have every cell of your body having a loss of the CCM gene, does it make your brain more permeable? And in fact, it does, even when you don't have lesions. So that when we measure the brain permeability of a 30-year-old with CCM1, 2, or 3 disease, in the areas outside the lesions, this is the normal brain, the brain that makes you think and talk and everything, that brain is as leaky as a 70-year-old. And we've shown that, and it's amazing, it's striking. So in the aging, it increases gradually every decade. But if you have the familial disease, you already have the background leakiness of a 70-year-old at age 20. So what does that really mean? The group from New Mexico, which studies families that have all the same CCM1 common Hispanic mutation, so that there are not many variations among the genetic variant of their disease, they found that patients with cavernous angioma, as they get older, have more stroke-like lesions in the brain for their age. So that all of us, when you look at an MRI, if you're 70 or 80-year-old, there are little areas of like incomplete strokes that are the wear and tear of aging. You get these 20, 30 years earlier if you have familial CCM disease. So that when I look at the brain of a 50-year-old with familial CCM disease, outside the cavernomas, just the brain itself, it has the telltale signatures of an older brain, or of a brain of a smoker or a hypertensive. So, you know, if you smoke and you have high blood pressure, you can accelerate this process, but you can also accelerate it if you don't smoke, don't have high blood pressure, but happen to have the CCM gene. This is important because I think as we get older, if we have the familial CCM gene, we have to be particularly careful about risk factors that can make this process worse. So smoking, high blood pressure that can make anybody get a stroke can make us more vulnerable to have that problem, aside from having the cavernous angiomas. Just our brain, as we age with CCM, is already predisposed to look more like at the molecular and cellular level, and at the leakiness level, like the brain of older patient than our biology would suggest. Nobody wants to hear this, but it is a fact, and it makes it worse if you have had a bleed from your cavernous angioma, because, you know, it's like an older person having had a stroke rather than a younger person having had a stroke. So it makes the ability for the rest of the brain to adjust for those for the effects of a bleed less plastic when you have this aspect going on in the brain. Now, thank God for the second copy of the gene, because you know, in evolution or through God's creation, the second copy does a lot to make up for the lack of CCM function. But if you really had a lot of loss of CCM function, the brain would be a lot worse, you know. So I think the idea here would be that if in the future we knew how to restore gene function, which, as you know, many people are working on this potential, if you restore gene function, you're going to protect not only the brain from forming cavernomas, which is also good, but also from this chronic leakiness that we have in the brain and maybe premature aging that people have. So when we studied CCM3 disease, and Connie knows this research, we found that many patients were having cognitive problems, some of them at young age, but some of them as they aged, and it was not related to the number of lesions they had in their brain. So you could have 100 lesions. It's obviously worse than having 20, but there could be issues with cognition that occur just because we have the gene in the brain that doesn't have cavernous angiomas. So learning disabilities, earlier cognitive problems are unfortunately something that we need to study more, and we need to understand more, and so forth. The cavernous angioma knowledge has also applied to the general aging, so that these groups of genes that happen in cavernoma that are affected by the microbiome, ADAMT, by thrombospondin, these are molecules you've all heard about. Those same genes are impaired in normal aging. So it is amazing that so maybe if there is a drug that helps cavernous angioma, it'll also apply to aging. And some people may know, if you do a lot of Wikipedia type digging, that you know, a drug like rapamycin was studied years ago as an anti-aging drug. It didn't pan very well there because a lot of side effects, and most people when they age, they don't need something to make them hyperglycemic on top of it or whatever. So people kind of abandoned it for that. But it had very good anti-aging properties. So some people thought that if we could keep the dose very low so that the side effects are non-existent, maybe it could prolong life by 10 or 15 years taking rapamycin. So the National Institute of Aging has done a lot of research on this. So now you're seeing rapamycin pop up as a treatment potentially for cavernous angiomas. So mTOR activity, which is the signaling that rapamycin inhibits, is also one of those signals that occurs as we get older. So before I go to the issue of iron and to the issue of plasticity, I want to just ask Abhinav to tell us a little bit how he is looking at this issue of microbleeds as we age, and what he's doing both in studying the microbleeds in the human brain as well as the microbleeds in the aging mice. So Abhinav, very quickly kind of tell us a little bit what you're doing in the lab, because I think people don't know about that and it may be interesting.
Yeah, thank you. Yeah, so the first step of what I'm looking at is when we look at a microbleed, what kind of cells are there, what brain cells, what cells that are normally circulating throughout the bloodstream, what is in and around those lesions. And what's very interesting is we see a lot of similarities, as Dr. Awad and Dr. Gerard have mentioned. We see a lot of similarities to CCM, which then, as Dr. Awad mentioned, so many of these genes, which these genes are usually at a certain level, but as we age, we're seeing certain genes go up, certain genes go down, which is causing this increased leakiness. So one of my first questions is, what are these cells there, and how are they relating to those problems via what genes in these cells are dysregulated, or which means they are different from the normal. From there, I'm looking at, as Dr. Awad mentioned, you have breakdown of this blood-brain barrier and how some of those proteins and microRNAs, these molecules from the lesion, gets into the blood, and can we measure that via a blood test? Because one of the most important things is being able to diagnose this and then predict in the future, is this person going to have a big hemorrhagic stroke? And by being able to access these molecules, we can create a blood test, hopefully in the future, similar to what we're working on with CCM, to be able to assess those.
So this basically follows the same theme that bleeding in the brain is always long in coming. So we don't wake up and the bleed happens. A bleed happens because stuff is weakening either in the cavernoma or in normal brain. So if you're older and you have a hemorrhage in the brain or a microbleed, that's because these blood vessels weakened over time, and there is an opportunity to detect in the blood the signals that preempt bleeding. And as you know, we're working on similar signals in cavernoma, whereby maybe within the next 5 years, we're going to have a test that will be very reassuring to people to tell them that the cavernoma they have in the brain is unlikely to bleed next year. And conversely, if that blood test does not reassure us, we will then maybe start some medications that otherwise we would use in people who bled to try to preempt the bleed. So I think this, or maybe even take the lesion out if one was sitting on the fence as to whether to take it out or not. So I think these are not just theoretical problems, they're very important problems that will help us understand what things precede and trigger a bleed. And it does seem that aging follows along a slower time course what happens with the CCM gene loss. You know, so statin is interesting, right? So people take statin to prevent having leaky blood vessels, and there have been some trials started to look at statin preventing cognitive impairment with aging. So that the idea here is that if your blood vessels leak less, then your cognition is preserved longer and longer. So the field of, they call it senescence, which is aging field, is filled with Rho kinase inhibition, rapamycin, mTOR inhibition, etc., and probably a gut-related effect as well, related to the aging brain and cognitive impairment being driven by the microbiome, similar to what we found in cavernomas. So the next couple of things I want to say before we open to questions have to do with this idea of iron. So we have studied the aging brain in people who don't have cavernoma and in the aging brain with cavernoma. And we find that, in fact, various areas of the brain accumulate tiny amounts of iron, even when you don't see a microbleed and even when you don't have a cavernous angioma. So there is some iron accumulating in the brain with every decade that we get older. It has not quite yet been correlated with cognition, so that it's a big leap for us to say that's what makes us impaired cognitively or not. So we do not know if that iron is bad for you and how bad it is. But similar to the areas of leaky iron around the cavernoma, there is iron in the areas of the brain that don't have a cavernoma as we get older. So that is something that was surprising to us, because the brain looks perfectly normal on MRI, but when we measure the amount of iron, it is in fact higher than in a younger patient per volume of brain. So that stuff is also being investigated by our group. So we talked about that, and the final issue is plasticity. So you know, the recovery from an injury requires the brain to activate circuits and to have neurons take over for other neurons, and the circuits to reconstitute themselves. So I'll give you a little example. If you had a bleed in an area near your speech area, you may be stuttering for 2 weeks, 3 weeks, and then you recover. So how do you recover? You recover because the area of the brain around the injury has basically taken over. And there is a lot of plasticity that takes place. And we know that exercise, for example, enhances motor plasticity. So if your arm is weak after a brainstem event, exercising it will allow you to regain some function. So when we study how that actually happens, looks like new circuits are taking over what was the injured circuit. So what happens there is it is extremely well known that with every decade of age, and this effect starts about age 40, so it's not really very old people. At age 40, your ability to deal with this recovery becomes slower. Just like people who are 60 years old don't find the word quite as well as when they were 40, the brain circuitry is not acting in the same way. And so many of you who have had lifelong challenges with the sequelae of hemorrhage or brain surgery, find out that as you get older, it becomes harder to deal with the same problem and to recover from it. And the seasonal fluctuations that used to give you some numbness every time the weather gets cold may get worse as one gets older because of this ability of the brain to react and cover up for injury. So it is real, and this means we have to be very careful about brain surgery in the elderly, because any added injury to the hemorrhage itself can cause a big challenge. But sometimes, obviously, it is necessary. So I think I tried to tell you about what we know about the biology of the problem, and both in aging alone and in the aging brain with cavernoma. And I know that each of you now are going to kind of say, well, what does it mean for me, and so forth. So let's use the rest of the time to answer some of your questions in this regard. So Connie, do you want to shoot them at me?
We absolutely have some questions. I think the questions that folks sent us ahead of time have already been answered in the context of what Dr. Awad has spoken about. So I'm just going to focus on the new ones in the Q&A. Please put them in the Q&A. Regarding high blood pressure, what would be an indication for high blood pressure treatment for a CCM patient? 140/90, 130/85? Is the top or bottom number more important?
Yeah, so that's a very, very good question. So for heart health, the bottom one is very important, because the bottom one is kind of the back pressure that your heart has to fight against. For the brain and the risk of bleeding, it's the systolic or the high number. So if the high number is high, that's what blows the gasket. The low number is what kind of is against the pump, against the heart. Both are important. Nobody wants to stretch the heart either. So I'm going to tell you what the guidelines are for the American Heart Association for blood pressure control, and many of you are going to be surprised, but actually normal blood pressure is in the 120s. Okay, so 130 is not okay, 140 is not okay. So the American Heart Association bases this on the fact that people who have 130, 140, 150 year after year, they have more heart attacks and strokes than people who keep their blood pressure in the 120s, even in their older age. So we no longer say, oh, you're a little bit older, your blood pressure of 150 is fine. That we don't say that anymore. So we say your blood pressure should be in the 120s, even if you're older, So I know I haven't changed my blood pressure goals as I've gotten older, and there is nothing wrong with being 70 and living with 128/85. Don't let it be 140 or 150, because that is going to cause a burden of disease over time, and that burden of disease is both heart attack, atheromatous plaques, and bleeding and strokes in the brain. So aside from cavernoma, blood pressure ought to be controlled to a tight level. The low one that's recommended is treat above 85. So again, treat doesn't mean take a medication. Treat may mean lower your salt intake and monitor it, exercise and monitor it. But again, I tell people that if you tried to exercise and lower salt diet and it didn't work, don't keep trying for 10 years. The burden of having that high blood pressure for 10 years is far worse than taking a small pill. And there are many, many excellent blood pressure medications now that have zero side effects. It's not like the ones that made you impotent and tired, and it's not like that. So there are a lot of blood pressure medications now that where when you do trials in 40,000 people, the side effects are the same in placebo as in the blood pressure medication group. So you can take them with almost no side effects. So this is a big pitch, both as a neurovascular doctor and as a cavernoma doctor for healthy blood pressure. Now,
Can I ask a follow-up?
yeah, I want to ask about does it make it worse for a cavernoma? Was that going to be your question?
No, it was going to be, does low blood pressure is there any concern about having low blood pressure for CCM patients?
Yeah, let me answer that first.
Okay.
The answer is no. So low blood pressure is only a problem if you have kidney failure and if you have coronary artery disease and narrowed vessels. So nobody is advocating living with 80/40. Okay, nobody wants that. So we want you 120. You know, it's good to have blood pressure, but it's not good to have it high. So low blood pressure will cause harm, will cause dizziness, will cause other side effects when it goes maybe 20 points below the normal. So when you start going below 100 on the systolic and certainly below 60 on the diastolic, you start having these symptoms of tiredness, dizziness, and they can be much more serious if you have a narrow artery, like a narrow coronaries, narrow carotid, etc. It doesn't harm your cavernoma that we know of, but to have lower blood pressure, but you're going to have harm from other things first. But high blood pressure has been associated in several studies with greater cavernous malformation hemorrhage. So in all the population studies where there was this data collected, sometimes you don't have that data collected, so it doesn't look like it's a factor. But in all the studies that I've reviewed where careful blood pressure was collected over time, the high blood pressure makes your bleeding risk from cavernous malformation higher. We have had several cases where there was what looked like a hypertensive hemorrhage, meaning a big bleed in the brain from high blood pressure in a patient who had high blood pressure. And guess what? In the middle of that hemorrhage was a cavernoma. So that person, yes, had high blood pressure, yes, bled from high blood pressure, but bled from their cavernoma when they had high blood pressure. So it is a weaker vessel than our other vessels in the brain, and it is more vulnerable to hemorrhage with high blood pressure. So it's another reason to keep your blood pressure controlled.
Okay. I'm going to shift to a different topic, genetics and genetic testing. Should people, there are several different questions, so I'm going to try to consolidate them. The question is genetic testing a good idea for everybody? Now that you're saying particularly as people age, knowing whether you have familial disease seems to be more important. So does this raise the value of genetic testing for everyone?
No, because there have been several studies, including one where I wasted a lot of money doing genetic testing in solitary cavernomas with zero yield. So we have evidence that you're going to, if you do genetic testing to look for CCM genes because you have a solitary lesion and no familial disease, you're going to waste your money. You're not going to find familial disease in those cases. So I had a grant for like almost $200,000 to look for, that's when I was working with Judy Galt in Colorado. We were trying to look for maybe the people who have a solitary lesion have familial disease and they don't know it. And actually that's not the case. So if you have a venous anomaly with a solitary lesion or multiple lesions around the venous anomaly, you're not going to have the CCM1, 2, or 3 problem unless you have multifocal cavernomas elsewhere in the brain. So yeah, you can have a vein and many other lesions that would be CCM 1, 2, or 3. But if all your cavernomas or only one is next to a vein, you're not going to have the CCM 1, 2, or 3. So don't waste your money on that. A better test would be eventually to look for the blood signs of a PIK3CA somatic mutation, because you may have a somatic mutation in that area of the brain, and we may ultimately be able to detect it in the blood, but that's different from the inherited familial disease.
Okay, thank you. To the blood. If iron accumulates in the brain, can a blood test tell us if our iron in the brain is elevated?
Indirectly. So if you have iron in the brain and you have some inflammatory signals related to either the cause of the leak or the effect of the leak, you may detect that in the blood. So the way we see it is, for example, as you get older, some tests called the C-reactive protein and tests called VEGF, which are angiogenesis and inflammation, are higher, and they correlate with the amount of iron you have in the brain. But is it a cause or an effect? Did both of those cause the iron, or did the iron cause those blood tests? So the fact of the matter is that, yes, there will be blood tests to tell us how our brain is aging, and they may be similar tests to those we're developing for cavernomas.
Okay, thank you. So if a CCM patient has mild cognitive impairment after hemorrhage at any age, is there a thought that they may then develop dementia faster later, but earlier than the general population?
We do not know, but we do know that people who have had prior brain injuries age, their vulnerability to cognitive change in the brain is higher. Individual variation is huge. So there are people who have one focal injury from which they recover, and it will not affect at all the rest. But if people had 2 or 3 brain surgeries, there was a lot of trajectory of different areas of the brain injured, and you maybe have a lot of lesions, yeah, then it may make the person age a little bit more. So we have this has become a focus in our clinic. When I meet somebody over age 60 with familial CCM, it's a big factor. Unfortunately, other than treating the CCM healthy brain and treating general risk factors, there's nothing else we can do. So what we tell people to minimize the cognitive effect is what we tell everybody to minimize cognitive effect of aging. The biggest factor so far identified, including in big clinical trials, is exercise. So the more you exercise, the less demented you will be as you get older. There's no question about it. The other issue is vitamin D deficiency. That can make cognitive changes a challenge. So you're getting a theme, right? We're telling you the same things we tell you to have a healthy CCM brain are the same things that will probably help you deal with cognition.
And someone else has asked, and so achieving and maintaining the health of our gut microbiome, will that be impactful?
Yeah, so the gut microbiome studies in Alzheimer's disease showed a different microbiome link than the ones we found in CCM. So the microbiome that makes you bleed from CCM is very specific to gram-negative bacteria, and even more closely to the bacteria generating a molecule called lipopolysaccharide. So that is something gram-negative bacteria, when we have a UTI or when we have prostatitis, if those bacteria increase in the gut, then we have more of lipopolysaccharide in the blood. And that is what drives bleeding from cavernous angiomas. And we showed that with Dr. Khan very elegantly by eliminating some of the genes, and that is really the effect. So it's a very, very specific effect. It's not the same thing with Alzheimer's microbiome. And it's not the same thing with Parkinson's microbiome. So when you look at the permissive microbiome of Alzheimer's, it's not the same permissive microbiome as cavernous angioma. So we have a lot more to learn about what is a healthy gut. And that's why I tell people the answer to it is not just to take antibiotics or probiotics hoping you're going to change your gut. It's not like that. It's a complicated balance of bacteria, and it may be different for some diseases. The only thing we know for sure is we don't want gram-negative ones to be overtaking for cavernomas. That we are sure of.
Okay. I'm going to switch topics a little bit as we just have a few minutes left. Can you update us a little bit on the Atorvastatin trial?
Yeah.
So actually, the question here that someone asked, but I think it opens the broader question is, does the doctor recommend statins for those who have bled?
So the issue of bleeding is complicated. So there is some concern in the general aging world that people who have had a hemorrhagic stroke sometimes were more likely to have been on statin. This signal was detected in a couple of studies among 30 studies or so. We're talking about studies that includes hundreds of thousands of patients taking statin. So many of those studies showed no signal, and it was never the primary outcome. What we do know now is that if you need it for your heart or for your cholesterol, you should be on it even if you've had a hemorrhagic stroke. So that is the fact. There is one trial being done currently by the NIH that has to do if you had a massive brain bleeding and you were already on statin, should you restart it or stop it with regard to having other bleeds? But that's not what you all are worried about. You know, that will be settled with this issue about big hemorrhagic strokes. But for cavernomas, the way statins may work, and I say may because we're doing a trial, and the way they work in mice is they decrease the leakiness of the cavernous malformation, decrease the iron, and decrease the bleeding from the cavernous malformation. So in the mouse cavernoma, the ones that are on statin have less iron around them, less bleeding, and the lesion is smaller over time. That is what made us go to the trial. You know, that's exactly what made us go to the trial. So what we are seeing so far in the trial is that there is no signal effect of a problem of safety, so that everybody in the trial has bled, or we wouldn't put them in the trial. It's a cash trial. So you already had a hemorrhage from your cavernoma. When we put half of you on statin and half of you on not statin, the ones on statin is not totally shutting the bleeding, but it's also not increasing the bleeding in a huge way enough to stop the trial. So we have safety reviews all the time that are looking at this, because I am blinded and you are blinded, so you don't know what you're taking. But we have no concern that it's making you bleed significantly more until we complete the trial, and we may see a difference there. We hope the difference will be the other way, where it is helpful, and then we will tell people you're more beneficial to be on statin. I just can't tell you right now. Now, what I can tell to the person who asked the question is that if you had hemorrhage in the brain from cavernoma or not from cavernoma, if you need statin for your cholesterol, for your heart, for all the other indications, yes, a bleed in the brain doesn't mean you should not be on statin. So, and I do brain surgery with patients on statin. We give it to aneurysm patients all the time. So it doesn't like make you, there is no reason to stop statin if you had a bleeding in the brain, if you need it otherwise. But I cannot tell you you should take it for bleeding. That's what we're doing the trial to find out.
Okay. All right. Let me get back to I was scrolling down and I lost the question. Are there any blood pressure drugs that you would specifically avoid if you're a CCM patient?
No, I mean, there is some interest in the beta blockers, which is a group called propranolol, metoprolol, etc. So those work by inhibiting the beta activity, makes the heart pump a little bit slower and a little bit weaker, and that's how they lower blood pressure. We find that that effect may have benefit on cavernous malformation in animals, for sure. In humans, we don't know how high you have to push the propranolol or metoprolol in order to have that benefit on cavernous malformation. So that's why trials are being done for that. But I think if you need a blood pressure medicine and they tell you to take a beta blocker, I would go for it. So that is fine. The problem happens with people who take propranolol and don't have high blood pressure, and then they lower the dose so much because they get dizzy when they sit up and whatever. So they have low blood pressure from those drugs, and that they don't tolerate that very well.
Mhm.
So if you have high blood pressure, they want to put you on a beta blocker, go for it. It may help your cavernoma too. All right.
Good. It's a twofer. All right. Now that, again, switching a little bit, we have about 4 minutes left. Now that we're 2.5 years into the pandemic, is there any new research about the impact of COVID infection on CCM, or are there any links between long COVID and CCM?
So long COVID, we really don't have, and it hasn't really been studied differentially in CCM patients. In our registry, as you know, Connie, that Connie helped us do with Kelly Fleming from Mayo Clinic, and Agnieszka did a big job with that, and folks from the Alliance. What we learned from that is that many patients were continuing to have symptoms of headaches and other symptoms late after they had a COVID infection with cavernoma. Now, I see that also in non-cavernoma patients, in aneurysm patients, and disc disease patients. So we're not really sure if the cavernoma affects long COVID more or not. The only thing we did see that was very compelling is that cavernomas, particularly the ones with a vein, are very likely to bleed if you have an active whopping COVID infection. It hasn't been the case as much with the Omicron because, many people are getting COVID. They don't even know they have it, and so it's hard to tell who bled from COVID, who didn't, but we're not seeing these very compelling hemorrhages, except if your COVID is very severe. So if you're not vaccinated and you happen to get one of those very, very severe COVID infections, it can cause bleeding in the cavernoma. We recently, in fact, our last patient in the Atorvastatin trial that we enrolled had bled in COVID, and then she was enrolled in the Atorvastatin trial. So COVID is not good for your cavernoma, so try to avoid it.
Okay, get vaccinated and boosted.
Yes.
That's
and it'll help both long and short COVID.
Yes.
It'll help all COVID. That is good. Anything burning that you saw that we didn't address?
No, we have questions, and I'm sorry, there were a lot of questions that haven't gotten answered, but I think related specifically to the topic that we're addressing tonight, I believe the questions in the Q&A are answered. The extra questions in there, we will try to get back to all of you with the answers that we have.
Since we have 1 minute, I did want to answer, sorry to interrupt you, about what's going on with the Atorvastatin trial, because I never got to answer that. So we have enrolled everybody. So we no longer are enrolling new patients. So the trial is closed for new enrollments. And we are following the patients over the next remaining year and a half. So because the last patient enrolled need to be followed for a full 2 years. So that's when the blind will be unblinded and will see the light. All right.
Okay. All right then. So we're at the top of the hour, and I want to thank all of you for taking the time to be here tonight, both as attendees and definitely as panelists. Darla has dropped in the chat the link to support group if you'd like to attend and talk some more amongst yourselves about these topics. She'd be happy to see you and have a chat. So thank you, Dr. Awad. Thank you, Avi. Thank you, Agnieszka, whom we did not hear from and is not feeling well today. And thank you, Ramul, and the baby too, because she's really cute.
Yeah, really thank you everybody on the team and you, Connie, for keeping everything going and for focusing us on the right questions and answers. And this is a process and we'll keep at it. I look forward to seeing everybody next month at or as many of you as we can at the CCM meeting.
Yeah, well, and let me remind people, there is a patient meeting at our first national patient meeting since the pandemic in Durham, North Carolina on November 18th and 19th. Registration information is on our website and it's all over social media. Just go ahead and find it. If you can't find it, write me. I will get it to you because we'd love to see you there. Okay, thanks everyone.
Thank you all. Bye-bye. Thank you.
Bye.